I'll be talking about blue carbon monitoring in the Pacific, along the Pacific coast, and also about a database that we've compiled to really pull together all the information that's been gathered by other speakers in the symposium, as well as some of you on the call and lots of researchers along the West Coast. So I want to provide a few additional background slides. We've had a lot of background material from Melissa, Laura, and John so far, but I'll kind of reiterate a couple of points. And then I'll introduce everyone to the Blue Carbon database. I'll talk about a regional data synthesis that we accomplished based on the database, and then give you a little bit of information about how to access the data if you're interested in blue carbon for values for your specific region. The last part of my presentation will be on monitoring blue carbon functions. So I'll go over how to monitor stock sequestration rates and greenhouse gas emissions. A little bit of that will be a repeat of the other speakers, but hopefully will provide some valuable information if you're interested in monitoring blue carbon in your ecosystems. All right, some background. So this is a fairly busy slide, but this kind of summarizes the various blue carbon processes that occur in tidal wetlands, whether you're thinking about eelgrass, marsh, tidal swamp, etc. The four blue terms are sort of the key ones that we want to think about when we're thinking about understanding all of the blue carbon processes. So we have, for example, on the very bottom, we have our stocks. This is the long-term storage of carbon in these ecosystems that we often measure with cores. And by long-term, we usually mean century to millennial scales. In the upper left, we have carbon sequestration. So John talked quite a bit about this, but this is the new carbon that's coming into the ecosystem. Some of that is due to photosynthesis by the plants themselves. So eelgrass, marsh plants, tidal forests. Some of that carbon is also coming in as flux from the watershed. So this is carbon fixed elsewhere, but it's being deposited and then stored eventually in tidal wetlands. In the middle, we have greenhouse gas emissions. So this is the dark side of blue carbon, because these are emissions from tidal wetlands that are negating some of their climate cooling potential. And there are three main greenhouse gas emissions that we want to think about in terms of blue carbon. So carbon dioxide itself, which is just plants and animals, microbes respiring carbon. Methane, which is a very potent greenhouse gas. So it has 30 to 40 times the heat trapping efficiency of carbon dioxide in the atmosphere. And then the last is nitrous oxide, which is a really potent greenhouse gas. But fortunately, in many tidal wetland systems, measurements conducted so far suggest that it's pretty much a trace gas and often not that much of a concern. So of these three greenhouse gases, methane is really kind of the biggest concern. The final blue carbon component is on the right of the slide, and that's lateral export. So this is the loss of carbon from systems, and this is probably the aspect of blue carbon that we have the least information on, the most data gaps. Okay, this was covered a little bit before, but where is blue carbon stored? So if we're thinking about stocks, thinking about new carbon coming in, we have a couple of different carbon pools in most of these wetland ecosystems. We have the above ground plant biomass, the below ground plant biomass. We have dead plant matter, and that can be wood, and even marshes will have wood that comes in from the watershed, and then the soils. And it turns out that the vast majority of the carbon stored in these ecosystems is found below ground. So mostly in the soils, but also in that dead plant matter. So these are just some summary figures here that you can see, even in a tidal swamp, which has lots of great above ground woody biomass, about 80% of the carbon is stored below ground. Okay, Laura introduced us to a whole bunch of these ecosystems, but I'll just refresh your memories. These are kind of some of the classic blue carbon ecosystems along the Pacific coast, and then a couple of additional less classic, but interesting and up and coming blue carbon ecosystems. So we have kelp forests in Southern California and Central California. Those are giant kelp forests. From Northern California up to Alaska, we have bull kelp and Nereocystis forests. These, there's a lot of interest in kelp forests as blue carbon ecosystems, but not a lot of data. And as Melissa described early in her first presentation, the mechanisms of how kelp forests are storing carbon are likely different than a lot of these more estuarine environments. We have tide flats. These are unvegetated, but these can be quite expansive in a lot of estuaries on the Pacific coast. And I'll show some interesting data. John alluded to this a little bit already. We have the seagrasses, marshes, and as kind of the classic blue carbon poster children. And then in the Pacific Northwest, we have these scrub, shrub, and forested tidal swamps. Laura talked about these in good detail, but these are increasingly rare ecosystems that store a lot of carbon. We don't have a lot of data on them, but there's, I think, a growing momentum to think about conserving the remaining tidal swamps and then also restoring these ecosystems. So we developed a blue carbon database. This was initiated in about 2016 with a grant from the Neuroscience Collaborative. We were, in that particular project, we were collecting a lot of our own blue carbon stocks data from Northern California up to Washington, but we also wanted to begin compiling a database of the work that other researchers have done on the West Coast. We started in the Pacific Northwest for a couple of years, and I work a lot in California as well and kind of interested in the whole Pacific coast of North America. So we expanded the database and started to pull in data from Mexico, California, Northern British Columbia, and Alaska. The database mainly compiles at this point in its existence carbon stocks data, but we're growing the carbon sequestration and the greenhouse gas components of the database. It exists as a series of linked flat files. So this is my very basic schematic of what that looks like. So we have flat files that are developed for different hierarchies of the database, starting with the different data sets. And then we move on to the sample metadata. A sample would be a core or a point measurement of greenhouse gas emissions, for example. For the cores, we have down core profiles, because as John showed, we often section these cores vertically to understand carbon accumulation rates and carbon density. So we have data on the core profiles. We have a separate section on accretion and carbon sequestration, and then a greenhouse gas flux piece of the database as well. Now, we're not just interested in carbon. I mean, carbon's cool for its own sake, and gas fluxes are cool for their own sake. But to really understand ecologically what's driving these patterns, it's important to collect environmental data. So when available, we've compiled data such as wetland elevation, soil pore water salinity and pH, the grain size of the sediments in the core samples, the surrounding vegetation communities, what plants are present, soil nitrogen, temperature, and groundwater level. So we've added that information into the database where it's been available. This is just one example of one of those flat files. It's been a beast to pull all this together over the last 6 to 7 years. But at the core level, we are approaching, I guess eventually we'll reach 2,000 cores, which is exciting for our coast. And at the subsample level, we have well over 30,000 lines of data, and that's what this screen capture kind of shows. Now, compiling all of this data, I think it occurred to us a couple of years ago that the database can serve a lot of functions for a lot of different end users, but as researchers, one thing we'd really like to do with that is to synthesize the data up and down the coast and try to assess some patterns. So we had two goals with this synthesis. We focused just on the carbon stocks piece at the moment. That was plenty. The first goal was to compare the stocks between different kinds of blue carbon ecosystems along the West Coast. And we looked at 5 of those ecosystems. And Laura gave you a preview of those results, and I'll talk about them a little bit more. And then our second goal was to really look at the relative influence of different ecosystem drivers. So things like elevation and salinity and plant community. But do these have a predictive capability when we're looking at carbon stocks on this broad geographic scale? So as Laura shared, we compiled 69 data sets. These were just least disturbed wetlands. So for this analysis, we ignored restored or disturbed systems. And this comprised almost 1,300 cores, or in a few cases, soil pits. That's how they do it in British Columbia. Apparently, they dig an entire soil pit, but you get the vertical profile of the data. We had one of the big challenges of the database was standardizing all the various methods that people used to sample these cores and also to do the analysis. Some people looked directly at percent organic carbon. Some people looked at percent organic matter. So we developed relationships. That's what this bottom figure shows for each of these different kinds of tidal wetlands so that we could convert percent organic matter into a standard metric of percent organic carbon. We integrated the carbon density over depth, and we looked at carbon stocks to 3 depths, so about 1 foot, 30 centimeters, half meter, and 100 centimeters. And the sample sizes get smaller as you go deeper because not everybody samples soils to the same depth. These two top figures show a summary of the cores by state and province, so all the way from Alaska to southern Mexico. Most of the cores come from British Columbia to California. Pretty good representation from the southern province of Baja California. And the figure on the right shows the distribution of the cores by ecosystem type. So by far, most cores came from emergent marsh, pretty good sample sizes from mangrove and seagrass meadows, and decent but smaller sample sizes from tidal swamp and from tide flats. So these are the results that Laura showed. This is a very slightly different version of that figure. These are box plots. The median is represented by the horizontal bar, and the mean value is represented by the little diamonds. Carbon stocks is on the y-axis in megagrams per hectare. And then the x-axis, of course, shows these 5 different habitat types. So from tide flats, seagrass, marsh, mangrove, and tidal swamp. So the 3 panels show the different depths. I'm going to focus mainly on the 100 centimeter depth because this is kind of somewhat of a global standard of computing carbon stocks to 1 meter depth. And the results there show a couple of interesting patterns. First, which has already been iterated in the symposium, is that emergent marsh holds more carbon in the soils than tide flats or seagrass meadows, and that the woody wetlands, the mangroves and the tidal swamps of the Pacific Northwest hold the most. So they have very similar mean values, similar median values, and hold about 5 times as much carbon as the low elevation wetlands, the tide flats and the seagrass meadows. I think one of the fascinating things that came out of this analysis, and John talked about this a little bit before, is that tide flats have as much, maybe even a little bit more carbon than seagrass meadows. The Prentice et al. Paper that John shared shows that result for the Pacific Northwest. This shows that that result is consistent for the whole west coast of North America. So these tide flats, which can be really, really extensive in some of our estuaries, sometimes they might comprise 50 or 70% of the tidal wetland area of an estuary, are actually holding a fair amount of carbon as well. They've traditionally not been thought of as blue carbon ecosystems, and we don't know exactly how long they do store carbon. That's an unknown, but we know that there's a fair amount of carbon in the tide flats. One of the other things we wanted to take a look at with the database is to try to look at some of the drivers of carbon up and down the coast of North America. And this is important because it can help us estimate blue carbon stocks where data are missing. Maybe you have an estuary where you have no samples at all, but you have some information on the salinity regime or the plant type. And you could use data from nearby estuaries to kind of estimate your blue carbon stocks. One of the analyses that we did, and this was led by Scott Bridgham at University of Oregon, is we took all of the environmental data that we had associated with carbon stocks, and we put it into a large machine learning model called a boosted regression tree. I won't go into the details of that model, but it helps you partition the data and understand the relative importance of different drivers. So that's what these figures on the right show. The first figure on the top, panel A, shows all the cores for the entire Pacific coast. We had a little bit less data for places like Mexico, so we had to remove some variables. But we divided up these environmental factors into processes that are generally operating on local scale. So this would be the kind of ecosystem you're looking at or the specific estuary that you sampled. And then also factors that vary on regional scales. So what climate zone are you in North America? What ecoregion are you in? Are you in the Mediterranean ecoregion of California or the maritime region of the Pacific Northwest? The type of estuary, are you a drowned river mouth estuary? Are you a lagoon? And then latitude. So the kind of take-home message from this is that for the whole coast of North America, estuary was actually the most important factor. So this is a local scale factor. And things like ecoregion or estuary type actually had explained very little to zero variance in the data. We also conducted this analysis for just the cores in the United States, minus Alaska, because we had better, I think, we had better elevation data that we could convert into tidal datums, for example. And then we had some PRISM model data that showed precipitation and temperature. But the same general result held, which is that the local scale factors, so ecosystem, the elevation of the wetland, and then the particular estuary that you're in, explained more variation than some of these very broad regional scale factors. So let's look at a couple of these factors and see what they say about blue carbon stocks. So elevation fell out as one of the important factors in the model. So we divided up the cores into four wetland types. We did not have elevation data for the mangroves in Mexico, so that was excluded. But we looked at tide flats, seagrass meadows in the low intertidal, and emergent marsh and tidal swamps, and kind of usually the upper half of the tide frame. Now, for two of these ecosystems, we found a positive relationship between elevation and carbon stocks. It's fairly weak for seagrass meadows, a little bit stronger for emergent marsh where we had more samples. These R-squareds are not great, so there's clearly a lot of variation unexplained by elevation. But in the case of marshes, for example, it suggests that marshes above mean high water, these would be tidal marshes we think of as high marshes, that they have higher carbon stocks than low marshes. Another thing we looked at was plant composition. So for seagrass, we compared Zostera marina, which is the native seagrass, with Nanozostera japonica, that's the non-native species in the Northwest. Pretty different sample sizes here, but we did see a significant difference with more carbon stocks in the native than in the non-native. And this might be because Nanozostera has invaded potentially mudflats that didn't have a history of carbon stocks. I'm not quite sure the mechanism there, but a little bit of a difference. For emergent marshes, this was a challenge because we have emergent marshes with such variable plant composition from Baja California up to Alaska. I mean, species are so different. So I took sort of a functional form approach and divided up the plant communities into essentially classes that corresponded both with salinity and with the kinds of structural features that the plants had. So the left two box plots show freshwater tidal. So these are freshwater graminoid and freshwater mixed assemblages. The middle two show brackish communities. And then the two at the right show saline. So these would be typically communities in California and Mexico that are succulent dominated. And a lot of variability in the data here, but one result that popped out is that the brackish wetlands may be holding a little bit more carbon than either their tidal freshwater or more saline counterparts. For mangroves, we looked at four mangrove types. We looked at Avicennia, Laguncularia, Rhizophora, and then mixed assemblages. And Rhizophora kind of fell out. This is the red mangrove. Excuse me, kind of fell out as the having the highest carbon stocks. And then finally, for tidal swamps, we looked at Picea sitchensis forest, so Sitka spruce forest. We compared that to two other types, really low sample size in the middle there, but we had okay sample size for hardwood forests. These would be alders and other species, but really didn't find any major difference there. But sample sizes are low, and I think there's a need for more data on the tidal swamps. So this does bring me to just one slide about data needs, and we need more methane emissions from freshwater and oligohaline tidal wetlands. Laura Brophy shared some information on our recent methane study. It was certainly promising news for tidal swamps, but for the marshes, we really found a lot of variability in methane emissions at freshwater and oligohaline sites. So we do need more data and to kind of constrain our understanding of the variability in those ecosystems. We do need more data, blue carbon data on restored and disturbed tidal wetlands. What did not include those in the previous analysis, but there's certainly intense interest in understanding the blue carbon function of restoring wetlands. So we need more data to kind of support our understanding of patterns there. We, of course, need more data on the lateral fluxes of carbon. It's not an area of my expertise, but very few studies kind of worldwide are looking at that, but there are growing numbers. Aaron Peck has done a little bit of work on understanding the lateral fluxes or the origin of carbon in Pacific Northwest salt marshes, for example. And then we need more data from kind of these more non-traditional blue carbon ecosystems. Scrub shrub wetlands are very rare, very undersampled. Kelp forests need more work, algal beds, and even sandy beaches. All right, so if you're interested in aspects of the database, how can you access the blue carbon data? Well, we have a paper in press, so this will hopefully come out in the next couple of weeks in Global Biogeochemical Cycles. We have a data release that's already publicly available. I have a link in the references at the end of my talk, and so if you download that, you can go straight to the data release if you want all the nitty-gritty of the detail. If you want summaries by estuary, we've produced some supplemental tables that will appear in the publication, so you can download those. For methane flux data, we also have a data release and a second paper in press at Ecological Applications. So if you're interested in that, again, the link is at the end of the presentation. Many of our West Coast data sets, we've also tried to facilitate incorporation into the Smithsonian's Blue Carbon Atlas. So that's what this little screenshot is on at the right. The Smithsonian's been compiling blue carbon stocks and sequestration rate data for the whole world, essentially. And because they have a very nice, well-structured database, and they have a public-facing application here in the database, we didn't want to repeat the wheel just for the Pacific Northwest. So our goal really has been, as we compile new data sets or dig through the weeds of old publications and find data sets, we've been sharing these with the Smithsonian to make them publicly available. All right, so the next part of my talk, I just want to discuss some aspects of blue carbon monitoring if you're setting up a new project or you're working in an estuary where there's very little blue carbon data, for example. And one of the first questions you want to ask is, do you have the capacity to collect new data? So that we have, as a community, we have needs for new data, but not all projects or not all restoration practitioners are going to have the capacity to collect new data. If you don't, you can ask the question of whether regional data are available. And thankfully for the West Coast of North America, just because of the work that our team's been doing and so many people on this call have been doing, we do have some good regional data for different areas. And then if you're working in a region where the data are sparse, you can also default to global values. So for example, you can use IPCC values to understand the blue carbon situation in your estuary. So I've kind of illustrated these as sort of different tiers of blue carbon monitoring. And of course, tier 3 is kind of the ideal. Collecting local scale data is always the ideal, but it certainly requires time and a budget and may not fit all projects. So how do you monitor carbon stocks? Considerations for that are asking, what ecosystems are in your project area? Are you working mainly in marsh? Are you working in tide flats and seagrass meadows? Does your project involve wetland conversion? So are you restoring, let's say, a former tidal wetland, such as pasture land, into a tidal marsh or into a tidal swamp? So you're going to need values for those particular types of ecosystems. And how deep in the sediment column do you think that your project um, impacts are likely to be? If you're restoring a site, you may be really focused on the surface layer, the new carbon that's coming into the system. If you're worried about a degradation of an existing wetland, for example, you may want to know the carbon situation in the top meter of the sediment column, because effects of that wetland degradation could extend to quite deep. This is an example of using the database as well as just some global values for emergent marsh in the Coos Estuary. I just picked one ecosystem type and one system and kind of wanted to see what this would look like if you're using different tiers of approaches. So at the local scale, we have 39 cores in the database for Coos Estuary marshes, and there's about 270 megagrams per hectare of soil carbon to 1 meter depth. If we just use the regional, so if we took the average of the entire Pacific Northwest, so British Columbia, Washington, and Oregon, we get a slightly lower value, 223. And then if we just use a global median, this is all the marshes. I think this is a summary of something like 15,000 cores. Globally, we get a value of 231. So these values are not dramatically different from each other, but you can see that there is some variability. And of course, having local data is always nice if you can achieve that.
You have about 3 minutes, Chris.
Great, thanks. So how to monitor carbon stocks? Well, John went over this already, so I can be pretty brief, but we want to collect soil cores to at least a 0.5 meter depth. You section those soil cores and then you analyze them for bulk density and organic matter content or percent carbon. You would then multiply that carbon density um, uh, by the length of the subsamples, and then you would sum those to depth, and that would give you a carbon stocks value. So for carbon sequestration, I'll go over this pretty quickly because John covered this well. But again, you want to ask, what ecosystems are in your project area and whether your project involves uh, wetland conversion. And you may want to know something about how changing hydrology as well can affect your carbon sequestration. So are you restoring a site? Are you concerned about long-term sea level rise, for example? The way to measure carbon sequestration with cores, at least, is pretty similar. You take the kind of the same core. You can use the same core for both stocks and sequestration rate, and then you do radiometric dating, like John talked about. Radiometric dating often doesn't work in young restored sites or in disturbed sediments, so you can use other methods such as the surface elevation tables that John talked about, or these feldspar marker horizons. This little ruler actually here shows kind of a fun thing that I discovered. This is a restoring site in Washington that's about 5 or 6 years old at the time we went out there. And because of agricultural activities, the former surface of the tidal wetland was incredibly distinct. So you would sink about 4 inches down as you were walking around and you would hit this hardpan layer. So you could literally just put a ruler down into the mud and measure exactly to that very distinct hardpan layer, and then divide that by the number of years, and that gave you an accretion rate as well. All right, I'm going to wrap up with monitoring with greenhouse gas emissions. If your site is likely to have high methane emissions, for example, if it is freshwater or oligohaline, you probably want to think about methane. You may want to use default values, or you may want to collect your own methane data as well. And there's several ways to do that. So I've just listed a couple of them here over this kind of quickly. First, you can use regional or global values of methane for your ecosystem. Of course, the pro of that is that it's rapid and the lowest cost, but the data may not be specific to the wetland you're really working at. You can use field-based chambers or with a portable gas analyzer. These are great for small sites. They're portable and they're rapid, but they're moderately expensive and they're fairly time consuming to collect the data. You can use an eddy covariance flux tower. This is kind of the latest technology in gas flux measurements. You can get continuous data over relatively large footprints. If you have a large site, this can be great. It's really expensive though. The flux towers cost over $100,000 each. And finally, you can monitor environmental drivers and you can apply a statistical model that Laura talked about, which we've done in the Pacific Northwest. This is fairly low cost, but you do need that statistical expertise to apply the model correctly to your site. So these are just some pictures of those methods. These are chamber-based approach, and these are flux towers. There are several on the West Coast. A new one got installed in Tillamook Bay recently, which we're excited about. There have been ones that have been running for about 10 years in Sassoon Marsh in California, for example. And then the final approach that I talked about, which is you don't actually collect any gas flux data itself, but you collect data on groundwater level, salinity, and temperature, for example. And then you feed all these detailed data into the model, and this will give you an estimate of annual fluxes.
Chris, we're going to have to wrap up.
All right, sounds good. So I'm going to skip this because Colin's going to talk about Tillamook Bay a little bit. Just some final monitoring thoughts. Your blue carbon monitoring should be accompanied by good ecosystem driver monitoring data, and you should make sure that you incorporate reference sites into your design. This picture on the right is just a quick plug for a monitoring plan that we've put together for the Central Oregon coast. Just published that last year. It does include detailed information on blue carbon monitoring if you want to take a look at that.